Hi, welcome to Dynamic Coaching Center. Today we are going to see about enzymes. Enzymes are the basic component of the cell. They involve in many biochemical reactions and metabolism in our body. They actually participate as a catalyst and involve in many reactions in our body. So they are called as biochemical catalysts, which are essential for life. Enzymes are basically made up of proteins or protein-like molecules. They react on molecules called as substrates to produce products. They might be product directly derived from the substrate enzyme reaction or involves two-step process which indirectly produce product by the enzyme substrate reaction. They reduce the activation energy of biochemical reaction and fastens the reaction rate. This is why catalysts are very important in biochemical reactions and metabolic reactions in our body. Enzyme reactions have three elementary components. They are enzyme, substrate, and product. The basic reaction are enzyme substrate combines and produce enzyme plus substrate product, or enzyme plus substrate combine and produce enzyme substrate. They produce product at the final stage which releases the enzyme at later. The later denotes that the last stage of the reaction. Enzymes just react with the substrate and increases the reaction rate, but they do not involve in the reaction. There are few types of enzymes which I have denoted here. Basically, few are simple enzymes. They are mostly whole proteins. Enzymes are made up of proteins. So, all the enzymes contains proteins, but which type of protein and which reaction they are involved and which product they produce, that is a major thing in the enzymes. Second type is holoenzymes. These enzymes are proteins which have small organic molecules in them. And third type is metalloenzymes. In this, there is metal composition with the enzymes. And fourth type is apoenzymes. Enzymes with cofactor, that is protein which binds to the coenzyme to produce the haloenzymes. That is, the third stage product is produced in this type of enzymes. Fifth type is isozymes. These are also called as isoenzymes. They differ in amino acid sequence. So, enzymes are basically made up of proteins, and those proteins are basically made up of amino acids. So, these isoenzymes differ in amino acid sequence, but though they differ in amino acid sequence, they have the same chemical reaction which they involve as a catalyst. There is no much difference in their reaction rate. Sixth type is alloenzymes. These enzymes produced by different alleles of same gene. Actually, they differ in the alleles of the same gene. So, This is why their name starts with L. Seventh type is ribozymes. These type of enzymes involve as a catalyst in covalent changes in RNA molecules. So these enzymes are called as ribozymes because they involve the RNA molecule changes in the ribosomes of the cells. There is a universal concept of enzymes. It is all enzymes are proteins, but not all proteins are enzymes. We have to understand this concept clearly because all the enzymes are made up of proteins, but not all the proteins produced are to be enzymes. Some might be enzymes, some might not be enzymes. So, proteins which are born to be enzymes are involved in the reactions, and some are used as building blocks or some other purposes inside the body. So based on the catalyzed reaction IUBMB, that is International Union of Biochemistry and Molecular Biology, this society have been classified enzymes in six different ways. First, we are going to see about oxidoreductase. These are enzymes which involves in oxidation reduction reaction. For example, dehydrogenase, oxidase, reductase and catalase. These types of enzymes involve in the oxidation reduction reaction in the biochemical pathways of the cell metabolism. Second is transferases. These transferase plays a major role in regulation of cellular process like synthesis of DNA or RNA. For example, acetyltransferase, methylase, protein kinase, etc. These type of enzymes involves in the DNA or RNA synthesis because DNA or RNA synthesis requires enormous of enzymes in winding of DNA, unwinding of DNA, ligation, sticking, amino acid attachment, polypeptide elongation, etc. So each enzymes are involved in different pathways in cellular and cellular regulation of DNA or RNA. Next is hydrolysis. These enzymes involve in hydrolysis reaction where a molecule is split into two or more smaller molecules by addition of water. So here addition of water involves. This is why they called as hydrolysis. Water molecules are involved in this reaction. Example, protease, caspase, nucleases. In this nuclease, there are two types of nucleases, exonuclease and endonuclease. These proteases involve in protein production and degradation. Caspase involves in the tumor pathway, that is cancer pathway. Nucleases such as endo and exonuclease are named based on the reaction where they are involved, that is outside the cell or inside the cell. Next, we are going to see about lyases. Lyases are the catalysts. They involve in the cleavage of C bond, that is carbon bond. It might be CC bond or CO bond or CS bond or CN bonds. This might be based on hydrolysis or oxidation. Decarboxylase and aldolase are the lyases reactions involved in the catalyst reaction. Next is isomerase. Isomerase are the catalysts which involve in atomic rearrangement within molecules. Sorry, LENS are left here. Example, rotamase, protein disulfide isomerase. This is called as PDI in short. Epimerase and racemase. The last is ligases. Ligases are the catalysts that join two molecules. For example, peptide synthetase, amino acid tRNA synthetase, DNA ligase and RNA ligase. So I have previously said that transferase involves in the synthesis of DNA or RNA. So here the transferase initiates the DNA or RNA synthesis, and this ligase involves in the DNA ligase or RNA ligase, because the DNA and RNA cellular process involves numerous steps in their duplication or replication pathway. At the final stage, after the amino acids, peptides, they are formed, after the polypeptide chain has been formed, after the new strand DNA or RNA strand has been formed, they have to be stick together to wind and make them as proper DNA or RNA. So these ligase, DNA ligase or DNA ligase involves in the DNA winding, RNA ligase involves in the RNA winding. This amino acid tRNA transferase involves in the tRNA synthesis of the amino acid production. So finally, EC, enzymes commission number. So we have been introducing or finding new enzymes that has been produced inside the body or outside the body. I mean in the laboratory, in vivo or in vitro. We have to be identify them based on their numbers. So there is a nomenclature, and based on the 6 types of enzymes, basic enzyme classification, they are numbering or naming the enzymes produced. For example, a new enzyme has been produced and it has been named as EC1, 0.11, 0.1, 0.6. So how this has been named? The EC denotes enzyme commission number. This one denotes the enzyme class, and next number denotes the subclass, and the last number denotes the subclass. So if it is 1, it denotes the oxidoreductase. If it is 2, it denotes the transferase. If it is 3, it denotes the hydrolase. If it is 4, it denotes lyase. If it is 5, it denotes isomerase. And if it is 6, it denotes ligase. So, based on this, they have been classified and named, and they have been denoted based on this number. So, this enzyme class is going to be the basic for the further classes, and further enzyme inhibition, Michaelis-Menten equation, Lineweaver-Burke plot going to be our next class sessions. Thank you.